Welcome back to our session on compartments and organelles. In the last part of this lecture, I would like to focus on the individual organelles present in our cells. So what you can see here is a list, or basically schematic view, showing you all of the organelles present in our cells, and there are quite few of them. We have the central nucleus, which is surrounded by the endoplasmic reticulum, which comes in two flavors. We have the rough endoplasmic reticulum. There is also a smooth one. We have structures called centrosomes, peroxisomes. We have microvilli. We have the Golgi apparatus, plasma membrane, and also mitochondria. So there are plenty of those organelles, and of course I'm not going to be able to cover all of the functions and the details, but I will give you a rough idea what those organelles do and what the function for the cell is. And let us start with the probably most interesting organelle within a cell, the nucleus. The nucleus has a simple function of storing the DNA within a cell. As I mentioned in the genetics lecture already, DNA is mainly stored in the nucleus, but there is also a small amount of DNA in the mitochondria. The nucleus is in charge of not only storage of the DNA, but this is also the site where replication of the DNA takes place. This is the site where the DNA generates an identical copy of the genetic information present in the cell. Not only this, also the process of transcription takes place in the nucleus. This is the process where DNA is used as a template to generate a molecule of messenger RNA. Within the nucleus, again, the information is not only stored but also condensed to the chromosomes, and we will be talking about the chromosomes and condensation of the DNA in the dedicated lecture in the weeks to come. For now, please keep in mind the DNA is packaged as chromosomes. Chromosomes are quite long and histone and are usually accompanied by proteins called histones. Together, DNA and histones are referred to as chromatin. But again, we will be having a dedicated lecture on DNA organization in chromosomes later on. The next organelle we need to have a look at are the ribosomes. So once the mRNA has been produced in the nucleus, it gets exported, processed, and it docks to the ribosomes, where polypeptide chains are produced based on the sequence of bases within the mRNA. Ribosomes are composed of RNA and protein, and usually there are two subunits. We have the small subunit and the large unit of the chromosomes. Both of them play slightly different role during protein biosynthesis, but we'll be talking about this later on. They carry out the protein synthesis at two locations within the cell. They do it in the cytosol as free ribosomes. However, most of ribosomes are present on the outside of the endoplasmic reticulum. Those are bound ribosomes. So how does the endoplasmic reticulum look like? The reticulum is a network of membranes or membranous tubules within the cytoplasm of a eukaryotic cell, and they are attached to the nuclear membrane, what you can see here quite nicely. So most of those ribosomes producing proteins can be found there. The endoplasmic reticulum, however, also does have a second form, namely the smooth endoplasmic reticulum. There are no ribosomes involved, and the function of those smooth endoplasmic reticulum membranes is carbohydrate metabolism, and also to a certain degree drug metabolism. So they are not necessarily that unimportant. They also play a role in ion movements, especially calcium. Many of our cells actually utilize calcium as a so-called second messenger, and we will learn about cell signaling later on as well. But for now, please keep in mind that the job of the smooth endoplasmic reticulum is carbohydrate metabolism, drug metabolism, and also ion movements. In contrast, as the name already suggests, the rough endoplasmic reticulum is not only associated with the nucleus, but also is full of ribosomes, and those are those tiny yellow dots on this schematic view of ER attached to the nucleus. And this is the site of the translation of protein destined for membranes or secretion. So what happens to those proteins that have been produced on ribosomes? They bud off from the ER in vesicles and migrate to the so-called Golgi apparatus. As you can see here, the Golgi apparatus is quite close to the endoplasmic reticulum, so the distance is actually not that large. So once a protein is produced on the ribosomes, they migrate embedded in vesicles to the Golgi apparatus. The Golgi apparatus has the function of packaging and shipping of proteins within the cell. So we can refer to it as a packaging and shipping station of the cell. So the molecules come in vesicles to the Golgi, fuse with the Golgi membrane, and then molecules can be modified by this very apparatus. After the proteins have been modified in the Golgi, they are pinched off in separate transport vesicles and leave the Golgi apparatus. Afterwards, those vesicles may combine with plasma membrane to secrete contents, and this is actually very important mechanism of cellular communications. Our cells do communicate, they talk to each other, and this happens mainly via molecules secreted in this way. Another extremely important organelle in eukaryotic cells are the mitochondria. So why are the mitochondria so important? Well, you might have been told this, but when someone asks what's the most important thing you remember from school, the answer is gonna be quite likely that the mitochondria are the powerhouse of the cell. And indeed, that this is the role of those organelles. Mitochondria have their own DNA. This is something I mentioned before. There is not a huge amount of DNA present in the mitochondria, but still they do have own DNA, and it is bound by a double membrane. Mitochondria break down organic compounds, including glucose, but also fatty acids, by aerobic respiration. So they do produce energy, and they do require oxygen. And the energy produced by mitochondria is mainly in form of adenosine triphosphate, short ATP. And please do remember that ATP is one of the most important molecules in our body. It basically keeps us alive by providing energy. But you will have also a dedicated lecture by Dr. Chro focused on the role of mitochondria in metabolism. The next organelle we need to look at are the so-called lysosomes. The role of lysosomes is aiding the cell in cell renewal because they contain digestive enzymes. They get rid of unwanted proteins. They break down old cell parts. They digest invaders, and this is especially important in cells of the immune system like macrophages. Good. Let's move on and have a look at the cytoskeleton. What are the main functions of cytoskeleton? The first important aspect of cytoskeleton's contribution to the cell is to provide the cell with certain shape and also mechanical stability. However, cytoskeleton is not a structure that is basically static. Cytoskeletons can be highly dynamic. This means that they can change shape very quickly, and this happens usually during processes like cell division, but also, say, migration. And this brings me to the next important function of the cytoskeleton. Cytoskeleton is actually critically involved in cell movement, and this is made possible by a dynamic cytoskeleton, so it can be rearranged fairly quickly. During cell division, cytoskeleton also contributes to equal distribution of chromosomes into each new cell during the cell division. So the functions of the cytoskeleton are quite diverse. Moreover, cytoskeleton also keeps the cells in place because it anchors the cells to neighboring cells and to the protein network on which the cell is sitting. It attaches the cell to the extracellular matrix. And the last contribution of the microtube of the cytoskeleton to the function of the cell is basically that they serve as railways along which motor proteins pull transport vesicles. As I mentioned before, some of the substances in the cell do require to be transported in an active way, and this is the job of motor protein. And those motor proteins do use microtubules that are parts of the cytoskeleton as railways. The main cytoskeletal elements are the microtubules, and they are made of two different forms of tubulin, the so-called alpha tubulin and beta tubulin. We also do have actin filaments, and actin filaments are slightly smaller than tubulin filaments that can be up to 25 nanometer large. In contrast, actin monomers are fairly small. They are in the range of they are in the region of 7 nanometers. Last but not least, we do have in our cells the so-called intermediate filaments. For example, neurofilament, which is specific for nerve cells. As the name already suggests, intermediate filaments are in between regarding their size. They are certainly smaller than microtubules, but they are larger than actin cytoskeleton, and they vary in size between 8 and 12 nanometers. So now I would like to show you a brief video summarizing the cell structures we have discussed so far.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
So, after watching this video and after maybe thinking a bit what we have learned today, you should be able to answer this question. So, which one of the following organelles recycles or breaks down worn out cell parts? Is it the cell membrane, ribosome, mitochondria, lysosome, or is it the endoplasmic reticulum? The answer is of course lysosome. I'm afraid there is not much you can actually deduct, and you need basically to remember this. So, which one of the following organelles manufactures proteins? Is it the cell membrane, ribosome, mitochondria? Is it the lysosome, or maybe endoplasmic reticulum? This is actually a quite tricky one. However, the answer is clearly ribosome, because answer E, endoplasmic reticulum, would include the smooth and rough endoplasmic reticulum, and only the rough endoplasmic reticulum contains ribosomes. Finally, which one of the following organelles packages proteins for the cell and sends them to organelles and other cells? Is it the cell membrane, Golgi apparatus, vacuole, lysosome, or is it the endoplasmic reticulum? Answer is clearly the Golgi apparatus, and again, this is something you need to remember. In order to help you remembering all those functions, we could actually think about the cell as a factory, with every single organelle doing different part of the job. In this model, the plasma membrane is the shipping and receiving department, because it regulates what enters and leaves the cell. The CO would be the nucleus, because the nucleus controls all of the cell activity and determines what proteins will be made. We have the cytoplasm, which is the factory floor and contains all the organelles, site of the most activity. We have the endoplasmic reticulum, which corresponds to the assembly line, because this is where proteins are being produced. We have the ribosomes, they do the job actually, and we have the Golgi apparatus, which prepares protein for use or export, and this corresponds to a finishing and packaging department. Finally, we have the lysosomes that are the maintenance crew of the cell, and the cytoskeleton, which is the support beams, walls, ceiling and floors. And last but not least, we do have our mitochondria, that are the power plants of the cells and transform one form of energy into another. Before I close the session, let us briefly recapitulate what we know about the differences between bacterial and animal cells. So, regarding the cell wall, this is obviously absent in animal cells, but is present in bacteria. The plasma membrane is present in both cell types. We do have endoplasmic reticulum in animal cells, which is lacking in bacteria. Microtubules are not present in bacteria, but we have those in animal cells. The same holds true for Golgi apparatus, nucleus, mitochondria, and the chloroplasts. Regarding the chromosomes, they are indeed present in both prokaryotic and eukaryotic cells. However, in animal cells, chromosomes are multiple units and are composed of DNA associated with proteins. In bacteria and other prokaryotes, this is usually a single circle of naked DNA. Ribosomes are present in both, because obviously both cell types need to produce proteins, and lysosomes are only present in animal cells. With this, I would like to close the session and thank you very much for your attention.